Hello everyone, this is Dr. Susan A Zwyea from the Health and Medical Technology Baghdad College. Today I will be talking about the immunoprecipitation based technique. So the first thing we have to know, what is the immunoprecipitation? Immunoprecipitation, or we call it IP, sometimes we call it pull down assay, is a widely used technique to isolate a protein of interest from a mixture using antigen-antibody interaction. So the word immuno refers to the use of antibody, and the word precipitation refers to the use of pulling down specific substances from the mixture or from a solution. And usually we use this technique to study the protein characterization and to study protein-protein interaction. Protein, or the protein of interest, or the antigen that isolated by the IP can be analyzed using the SDS-PAGE or the Western blotting that Dr. Ahmed talked about it yesterday. And actually a necessary step, this is considered a necessary step to verify the purification, and it's very important to use different antibodies during the Western blot to verify pull down of the correct protein. So going back to the principle of the IP, in IP a primary antibody is added to the mixture containing an antigen or a protein of interest and incubated to allow the antigen antibody complex to be formed. So now we have the complex form. Subsequently, later on, the antigen antibody complex are incubated with the secondary antibody, which we usually be immobilized or coated with the protein coated beads, okay, agarose beads. That's allowed them to absorb the complexes. The beads are then thoroughly washed, and the antigen or the protein of interest can be eluted from the beads by acidic solution or SDS, sometimes by heating. Okay, so this figure can be considered an overview of the IP. So here we have our cells. So we prepare the lysate by adding lysate buffer, and after we get the lysate, what we do in the next step called the pre-clearing. In this step, what we use isotype antibody to eliminate the non-specific binding, okay, and then after that we add the secondary antibody, which is coded to the agarose beads, okay, and then we have the next step, which is the pulling down. So when we add the isotype antibody to our lysate, the complex protein, the protein or antigen antibody complex will be formed, and then we add the secondary antibody that's coated or bind to the agarose beads, okay. So we can pull down, so that will bind to the complex. So next step, we can pull down that the protein of interest that's bound to the primary and the secondary antibody. Pulling down, we do centrifugation step, okay, and get rid of the supernatant, and then we can isolate the protein of interest, and then we elute it and analyze it with the Western blot. So we have a few notes regarding the primary antibodies used for the IP. Basically, we have two types of antibodies: monoclonal antibody and polyclonal antibody. Regarding the background, basically the monoclonal antibody has very low background, which is good, while the polyclonal antibody has very high background, which is bad. Okay. Regarding the binding affinity, the monoclonal antibody, the affinity considered to be low, so we really should be picky and pick only the antibodies that have high affinity, because otherwise the complex will not be stable. The antigen antibody complex will not be stable, or maybe will not be formed. For the binding affinity, in case of the polyclonal antibody, which is good and high comparing with the mono. Regarding the stability of the antigen antibody complex, The stability considered not to be high, it's very low, and to solve this case, so we have to use several antibodies, okay, to get the multivalent binding. While when we use the polyclonal antibodies, it will be very easy to get the complex, and the complex will be very stable because it's very easy to get the antigen, or because it's very easy to get the multivalence. Okay. Here the slide showing the three different types of beads that we can use for immobilization, specifically for the secondary antibody. So basically here the agarose beads and the magnetic beads are commonly used. For the agarose beads, basically it has a pore, and through that pore the antibodies can diffuse and bind to the internal matrix of the beads, okay, and that's how it's provide a high binding capacity. For the magnetic beads, it's also sphere rounded, and it's very easy for handling, but there is two limitations. The binding capacity is not high enough, okay, and the magnetic beads are considered to be expensive, okay. The cost is very high if we compare it with the agarose beads. It's cheap. There is another type of the beads which is combination of the first two types, which is called the magnetic agarose beads. So it will be easy for handling, and it also have a very high binding capacity. Okay. This is a slide showing the comparison between the three different types of magnetic beads. So in terms of the diagram of the beads and the structure, we can see the three types are rounded, and the agarose beads are the largest one, the diameter around 100 micrometer, and the magnetic agarose beads in between, and the smallest one, the magnetic beads. The actual images, how it look like, usually the agarose beads look like this, it's white, and the magnetic beads it's orangish, and the magnetic agarose beads look like gray or dark black. Okay. In terms of the binding capacity, both agarose and magnetic agarose beads have very high binding capacity, while the magnetic beads has moderate binding capacity. In terms of the likelihood of sample loss, we have the probability of having the sample to be lost during the steps. It's very high when we use the agarose beads. We can lose the sample through the washing steps, while when we use the magnetic beads or the magnetic agarose beads, it's negligible, the sample lose. In terms of centrifugation, we have to do the centrifugation step when we use agarose beads, while it's unnecessary when we use the other types. And in terms of using the magnetic rack, for sure it's very important when we use the magnetic beads or the magnetic agarose beads, while it's not necessary for the agarose beads. Visibility of the beads, as we can see from the picture, those are very excellent and very clear, while this is very difficult or very poor visibility. Other features in terms of the cost, the agarose beads, the cheapest one, is inexpensive, while the magnetic beads it's expensive. Okay. And the magnetic agarose beads is in between, less expensive than the magnetic beads. In terms of the procedure, so here we going to the steps quickly, and then we go through each step in details. So as we can see in here, so after we getting our protein extract or the lysate, we're going to add the primary antibody to form the antigen antibody complex. And after that, what we are going to do, we're going to incubate the complex, okay, for at 4 centigrade, 4 centigrade or Celsius for 1 hour, and we can leave it overnight on shaking rotator. And this is the shaker rotator. Okay, this is the cold room, and we can leave it for 1 hour or overnight. So after we allow this complex primary or antigen complex to be formed, and we can add the secondary antibody, which is coated with the agarose beads or magnetic beads or a mix of them. Okay, so we can add it and incubate also at 4 degrees for 1 hour, or we can leave it overnight on rotating shaker again. After the incubation step finish, we can do washing. Okay, washing step. The pellet, and here you can see the pellet, the agarose beads by the centrifugation. Okay, and remove the supernatant by aspiration without disrupting the beads. So we have to make sure we don't disrupt the beads. Okay, we carefully remove this supernatant. Okay, we do what we do, and then we do the washing. Well, the washing step could be with the we can use the ice cold lysis buffer, or we can use the washing buffer, and we repeat this 3 to 4 times, and every time we're going to remove the supernatant and keep the beads. Okay. So now we have the protein of interest, but still the bead is still in there. So how going to get rid of the beads? We before we do Western blot or SDS-PAGE or Western blotting, we're going to add SDS sample buffer containing 2 mercaptoethanol and heat it, boiling for 5 minutes, so we can get rid of the beads, and we will be only the target protein of interest in the lysate. Okay, and then we can use it later for the Western blotting, and or we use it for the SDS-PAGE. We have to keep in mind the antibodies are still there. Okay, so we get rid of the beads, but the antibodies could be there. Okay, co-eluted, and should be taken into account when analyzing the data. Okay. So on this slide we have a few notes about the detergents. So in terms of detergents, we have to do some pilot experiments to adjust the appropriate concentration for detergent we use in the IP to reduce the non-specific binding. And for the protease inhibitor, as we know, the proteins and antibodies are subjected for degradation. So we need to use some protein degrading enzyme or protease like PMSF and the EDTA to minimize that possibility. And for the elution condition, we just keep in mind that we have to use when we eluted the proteins, the buffer we use should contain SDS and some reducing agent like 2-mercaptoethanol. Okay, and here I will show you some picture of a procedure how we do it, starting from the cell lysate preparation. So after we get the cell lysate, we going to centrifuge the cells in the mini microcentrifuge for 3 minutes at 13,000 RPM. Okay, as we can see in here. And the next picture is showing us after we get the our cell lysate, okay, from the centrifuge, we're going to remove the supernatant and keep the pellets, which what we need. Okay, and here what we have to do is resuspend the cells in lysis buffer, and we said it should have the lysis buffer is REPA, and should have the protease inhibitor, which is PMSF. Okay, here in this picture, after we adding the lysis buffer, we disrupt the cell using a few quick pulses with a vortex. So here's the vortex, and we just getting some disrupting the cells. Okay, And not only vortexing, sometimes we need to use syringe. Okay, so we aspirate the lysate a few times and aspirate and push until we get the cell pellet to be disrupted. Okay, and after we done with the lysates, okay, we keep them on ice for 10 minutes and incubate the cell lysates on ice for 10 minutes. And now we see this is ice box, and then we put the tube in here that contains the cell lysates. And after that, after an incubation on ice for finish, we going to centrifuge the lysate at 13,000 RPM for 15 minutes at 4 degrees. We go to the cold room. Okay, we use the mini centrifuge, and this is the condition where we use it for 15 minutes at this speed. Okay, and 4 degrees. So after that, after the centrifugation finish, what we're going to do, we transfer the supernatant to a fresh labeled microcentrifuge tube. So here we're going to use a new tube and label it, and then put the supernatant and discard the pellets. This is the first step, which was the sample or lysate preparation. Now we move to the pre-clearing step. Okay, this is the second step. The first thing we need to do is we add the protein agarose plus agarose beads and the antibody. So we're going to add the beads and the antibody, a primary antibody, where to the lysates. Okay, to the cell lysates. So we start first adding the antibody, and then we have something very important we have to keep in mind, that when we're going to add the beads, do we vortex or not vortex? It's very important to vortex, okay, the beads before we adding them. Maybe so we make it's resuspended evenly. So we give a little vortex to the tube that containing the beads. Okay, we hit them on the vortex. Okay, and to make sure they're evenly resuspended. After that, what we're going to do, we add the beads to the tube that have the lysate and the primary antibody. Then going to add the beads. Okay, we do vortex for all three. No matter what kind of bead we use, we have to vortex first. And then after that, we incubate the lysate mix, okay, which contain the lysate, the beads, the primary antibody mix. Okay, we put them on centrifuge rotator, which this one. Okay, this is our tube that contains the mix. We put them in a cold room, which means 4 degrees. We leave them for 30 minutes, half hour. Okay, so after that, after this step finish, we do centrifugation. We use 3000 RPM for 30 seconds, very quick centrifugation, and 4 degrees. We're still in the cold room. Okay, after that, what we seeing here in this picture, we are transferring the pre-cleared supernatant. We take the supernatant again to a fresh labeled tube. So we are also going to label a new tube and transfer our supernatant to this new tube. Okay, and discard the pellets. So after that, we need to determine the protein concentration using Bradford assay, and I'm not going to talk about this. I will just mention very clearly, very quick. And so, you know, it's you prepare a few microcentrifuge. You put in each one 1 ml of Bradford agent, and then we're going to put. We use this. We can use this table or any table, okay, to add the protein, okay, of interest and generate the standard curve, so we can determine the protein concentration of our sample. And after that, we can read using the plate reader, okay. And generate, as I said, the standard curve in Excel and calculate the protein concentration, okay, of our lysate, of our samples, okay. So now we start with the most important step, which is the pull down. So for this, we're going to prepare two labeled tube or microcentrifuge tubes. One labeled control, the other one is test. Okay. The test will be, in this example, will be the C-MYC. Okay. And then we add the lysate plus the lysis buffer and make up this volume to 500 microliter. Okay, and then after that we are going to add to the C-MYC to the test and the IgG to the control. So now we have the primary antibody and the protein, and we're going to incubate them, okay, in the cold room for 2 hours to allow the complex to be formed. The next step, we're going to add the secondary antibody that's immobilized with the agarose beads, okay, and also allow the complex to be formed and incubated for overnight to allow this complex to be formed. After that, this step will be followed by centrifugation and the washing and multiple washing, and then and we carefully aspirate the buffer from the tubes and leaving the beads, which so we can later use the protein of interest. So now the next step is doing the SDS-PAGE electrophoresis, but before that we're going to add we're going to resuspend the beads in 200 microliter SDS-PAGE loading dye containing the reducing agent beta-mercaptoethanol and boil the sample at 95 degrees for 5 minutes, and follow that by centrifugation the beads, okay, at room temperature for 10 minutes, and later on we use the gel loading tips carefully, pipette the sample obtaining from the beads and load them into the well of usually the concentration of the gel we use it 4 to 15 gradient SDS gel. And then we do the Western blot analysis as yesterday Dr. Ahmed Maliki explained. So I'm not going to go through the Western blot steps. I'm going to talk about the results and how we interpret the results of the IP, which is a little bit different from the Western blot. It's similar but different. And in our example, we mentioned that we have two tubes, one containing the IgG, okay, the control antibody. This is labeled isotype control. This is the control antibody, and the other tube which containing the C-MYC antibody. So we labeled here C-MYC IP. This is the IP, okay, what's going to test. And this is the this is the lysate input. This is just the lysate without anything, okay, and this is the ladder, so we can know the molecular weights. So basically the C-MYC is usually show up below the 75 kD, okay, so this is the 75. This is usually we say this is the MYC. It's the molecular weight of the MYC about 60 something, 67, okay. And as we can see, the first line has no C-MYC, okay, because it actually doesn't have C-MYC, it has IgG, okay, while the C-MYC tubes show very strong band, which showing this is very specific, okay, so we can say that this is not specific bands, and those are the very sharp dark bands. This is the specific, and this is the C-MYC. This is something else, okay. And the question here, why the C-MYC didn't show up in the lysate input, and I put it here, because the protein has very low endogenous expression level that we cannot see the C-MYC here. Sometimes if we didn't use the C-MYC, we use something else, okay, if it show up here, it's supposed to show up here in the lysate input and the the test, our test should show up here and here, but if it didn't show up here, that means it has very low endogenous expression, but if it shows here, that means it's very high endogenous expression. I hope the presentation was clear and you get some new information, and I hope it was beneficial for everyone. Thank you, and if you have any question, just write down in the comments. Yeah? Thank you.